well about incretin class of therapies. What is the problem? I mean, most of the times at the onset of diabetes, it could be insulin sensitivity because the way we live today, it's a sedentary life. We don't have any physical activity. So the large amount of insulin is still there, but it's not able to dispose the glucose. So that's the main culprit. But you don't need medicines for that. Little bit of exercise, maybe at the most metformin would be sufficient. But we tend to forget that essential part of the life. You also need to have a physical activity, which is kind of a defect. And sometimes probably it's a dysfunction of the pancreatic beta islet cells. It doesn't produce insulin, and hepatic glucose overproduction. These are the three main reasons, but at the start of the disease, it's more of the insulin sensitivity or insulin resistance. And can you go to the next slide? So the earlier part of treating diabetes is to see that how you're able to produce insulin pancreatic beta cells. And if you have insulin resistance, you also need to look into the uptake of the glucose into skeletal muscles and the tissue, and how to stop the overproduction of glucose from the liver. This is the kind of pharmacological interventions to treat. And today, when a patient goes to doctor, it is not only the diabetes. The patient also has comorbidities. It could be part of type 2 diabetes plus cardiovascular, part of type 2 diabetes or obesity, or it could be 3 of that or 2 of that. So the doctor needs to see that the condition, what is the comorbidity. Whether you are having obesity, then you need a different class of therapy. It's not that simple sulfonylurea or something else doctor would be able to prescribe, because you need to be treated holistically, not only type 2 diabetes, but also in terms of the excess of glucose into producing the obesity, or in terms of cardiovascular things and all those things. So the treatment would depend on the patient, and in India, most of the times it's economy. So whether patient, even doctor would like to prescribe based on the condition, whether he would be able to affordable to have those medicines. So most of the times the doctor would prescribe sulfonylurea because it would come, or metformin at a cheaper price. And maybe it helps at the initial stages, but when you take some of these medicines, it goes there and it's there for 24 hours, and it continuously asks your pancreatic beta cells to secrete insulin, which it may not be required all the time. So it's essential that what kind of medicine is to be taken at the onset, depending on the HbA1c levels and what is the target you want to achieve that. The other aspect is that the life expectancy with all these comorbidities also decreases. If a person is obese, the expectancy of these age 70 with BMI of normal BMI, say less than 25 or so, it can be 80%. And if you go on increasing 35, 40 and above 50, the life expectancy could be diminishing to 50%. That's the problem. And though in India it's about 5%, but it's expected to be more on going forward. Next, please. So when you talk about the drug discovery, you would look into the selectivity. I mean, that's what we look into. You make a molecule, you see that you have a pharmacophore, and look into the biological assays and see that you have what kind of potency. Then you go for selectivity, then you go for absorption, metabolism, elimination, and you go for all the safety related things. And most of the times, I think the pharmacophore identification, probably you spend about 10% of the time. But that's one aspect. But the most important aspect is that the pharmacophore, when you test it, is in the isolated cells, in vitro systems. It doesn't look into the bioavailability, it doesn't look into the metabolism, it doesn't look into the absorption, it doesn't look into the tissue distribution or volume of distribution, all those things. So this is what I depicted here is about 5%, but you need to look into several aspects in terms of the absorption, distribution, metabolism, and excretion and toxicity. So when you go to the patient first, the first dose, you need to have the therapeutic window, safety window. At least you need to predict. If I say that I want to give this as a drug for the treatment about 10 milligrams, you should establish at least at the 100 milligrams dose, it doesn't have any of the safety related issues. So then you can start with 1/10 of the dose. So that's where we need to, but 1/10 of the dose will be very risky to start as a drug discovery program. You never would like to, though it's a regulators would accept you to start, but you never would like to do that. So at least you have 100-fold of selectivity in terms of the, what is the intended dose and the what you are looking in terms of the safety window. It needs to be such a large window. So that's what you would like, because what you are deriving from the cell-based assays and the animal models, which may not be relevant to the human at the end of the day. You think it would work in terms of whatever the correlations you have done, but may not likely to work. Next slide. So it's important that one needs to look into. I am coming to the actual thing. I mean, thanks to the Gila lizard, someone was looking into the toxin of this Gila lizard. And that toxin was found to be a peptide of 39 amino acids. And this 39 amino acid peptide also had some homology to the GLP-1. GLP-1 is an incretin class of molecule. And the curiosity is that if I see this contains a sequence related to the GLP-1, could it also be effective in addressing type 2 diabetes? Because GLP-1 augments the insulin secretion from the pancreatic beta cells. It's a natural way. If you take a food, and upon ingestion of food, and the food, when it is traveling from stomach to the GI, there are L cells which produces the this pancreatic hormone, and it goes and sits onto the pancreatic beta cells and stimulates the insulin, sensing the glucose levels in the circulation. That's the most important thing. It doesn't allow to secrete insulin unless there is certain levels of glucose circulation in the body. So that's the most important. It's a nutrient sensing hormone. And based on the nutrient levels, based on the glucose levels, this GLP-1 gets secreted and goes into the pancreatic beta cells and then augments the insulin secretion. That's a natural phenomenon. But what does it also has? It has a very short life. It's about less than a minute. So there is a dipeptidyl peptidase which goes and breaks this hormone and inactivates. So that's where the DPP-4 inhibitors became a therapy for type 2 diabetes, which is very benign, but it does produce a significant effect, benefits in terms of things, but in terms of cardiovascular, in terms of obesity, it doesn't have that much of a value, but it does produce. So you would like to see that how this toxin would behave as a GLP-1 agonist. So they found that there is a small change in the N-terminal part. Instead of alanine, it has a glycine, and it has some resistance to the DPP-4 enzyme. So its half-life is much better in terms of 2 minutes versus 2 hours. So you have a drug, potential drug with a significant improved half-life, but when they estimated the in vitro assays, it showed nanomolar potency of cyclic GMP assay levels. So that's the significant, but you know that it's a toxin. You should go to the regulators and say that I want to test toxin for treating a patient. That's the challenge. But still, I mean, as a scientist, you can find out what kind of toxicity, what is the target organ, how does it affect in the target organs, and what dose. And they could go to the regulators and convince, and this product got approved in 2005 as an exenatide. So that's the beginning of the therapeutic class, a toxin becoming a product of treating a diabetes in a natural way. So you don't need to be really looking at what I am doing, but as a scientist, you need to look how I am going to develop, what I want to understand, what I want to address, whether it gives a benefit to the patient, what kind of a benefit, and all those things. And next slide. So this is the exenatide, and this has only 53% homology. If you go to the biologist telling that 53% homology, he simply would throw away. I mean, you're talking about 53% homology and you want to make it as a drug. So that's a. But if you see today, the rest of the molecules, there are about several of the molecules from 2005 to 2024, 2023 approved, can have the. So this is the GLP-1, what I was discussing, a site which can be amenable for the DPP-4 cleavage, thus becoming inactive. So you can modify that amino acid to protect from those enzymes. And if you can look into, please, can I have that thing so that, okay. So this is where you would be able to modulate the sequence and it's not working. Modulate the sequence and make the analogs. Can you go to the next slide? Yeah. So these are some of the molecules. Exenatide was a twice a day product, then came liraglutide, you can take once a day product, then you got albiglutide and dulaglutide. It's once weekly treatment. And you have semaglutide, it's also once weekly treatment. Now you have tirzepatide and several other things. That's how the things are moving up. If you look into the semaglutide, the last year's sales is about 13 plus billions. One molecule in this class could produce 13 plus billions. And we talk about the pharmaceuticals in India is about 60 billions. So that's the innovation can create so much that you can be surpassing what you think. I mean, when you say that by 2047, 400 billion, is it possible to achieve? Is it possible to achieve? This company alone by 2030 could be 100 billion. This company alone. And our expectation to be 130 billions. As a country, the pharmaceuticals of 130 billion and one compound of this company could be 100 billions. So that's the strength of innovation. And how do we do that? Is it that we don't have knowledge? Without the knowledge, we couldn't have been the pharmacy of the world. So we have so much of understanding, so much of pharmaceutical understanding in terms of production, in terms of therapy, in terms of anything what you talk about. So there is a very much possibility we can do over the years. Next slide, please. So what we did is that we wanted to see that how do we understand this as a therapeutic class, what we wanted to do. As I said, that the most important thing, can we make a potent compound? So we have a next generation GLP-1 analog, which is about 80 picomoles. So you talk drug discovery in nanomoles. So what we are looking in terms of the product, a picomolar effect in terms of the cellular assays, cyclic AMP assays. And we have substantially a potent molecule. And if you look into what kind of next aspects you want to see that, so the biologically you can have several of the assays. When you say that you have the cyclic AMP assay by receptor, that tells about 87 picomoles or whatever it is. But you need to also see that how long this interaction with the receptor and ligand exists. So you need to look into beta-arrestin. If it doesn't, it's not a bias, then it gets internalized and degraded very fast. So that's another assay you need to know. That whether you did a partial agonist or full agonist, then you also make a GTP gamma assays. Then you will know that what kind of agonism you have and what is the receptor affinity. So you can also have the ligand, radioligand binding assay. It's not that one assay could be suffice to understand your molecule, but you need to have various other molecules depending on what you want to achieve in that molecule. Because when you say that one assay, I'm happy, I'm going to forward, but it doesn't work at all. You need to know that what is the affinity, you need to know that whether it's full agonist, partial, intrinsic characteristics. And the second aspect is that the CYPs is important. Unless you establish the CYPs, there is no induction or inhibition, you would not be able to take it forward. And there is a hERG potential, so the cardiac side effects. So you have to establish all the liabilities before you can put into the pharmacological understanding. Once you know that the pharmacological understanding, then you also need to know that what is this molecule in terms of safety aspects. Then you need to know at least 100 times or 200 times to that of the predicted dose. What is my potential target organs and how does it affect and what kind of things. And you also need to know that what is the half-life of your molecule. It's not that you're just looking a molecule just to give it, but if it doesn't have at least a couple of hours in circulation, it doesn't have any value. It could be significant potency, significant safety, but if you don't have some of these aspects of volume of distribution, all those things, it doesn't have it. So we need to do several of the things before you can nominate it as an IND candidate. So we did all those things, but more often you understand in lab, but you need to also do in a commercial scale. It's not that you isolate a molecule in solution, does it, all your experimental thing in a test tube and say that we have a molecule, but you need to synthesize on kilogram scales and multi-ton scales and see that your product is stable, see that you have product of flow properties, you are able to compress into a tablet or injectables or whatever it is. The physicochemical characteristics of solubility and your, several of the aspects, non-hygroscopic aspects and the polymorphic aspects and the crystallinity, flow properties, all those things are important. Unless you establish, you may have fantastic safeties, fantastic T-half and everything. If your product is not going to be pharmaceutically acceptable solubility or physicochemical properties, it doesn't become a drug. So that's the, yeah, okay. So, so we did in, sorry, maybe 10 minutes. Okay, okay. So I thought 40 minutes talk. So I thought 40 minutes talk. Okay. So this is the in vivo experiment in terms of the efficacy we compared with the standard of care products. And we could find at half the dose we have significant, the biological assays in terms of the in vivo assays. And of course, it's animal experiments. You would like to see that how does it, translates into the human trial. We did a study in obese subjects because we not only looking for the type 2 diabetes, we also looking for the weight loss. And when you go into the human subjects, you go into the healthy subjects. They are the part of the healthy studies of obese patients, and you see most of them are 35 to 40. And what we did see is that with one single dose, at a dose of 2 milligrams or 2.5 milligrams or 1.5 milligrams, we could see about 2 plus kgs weight loss and as much as 3.3 kgs on day 8. So we dose today and next Sunday you check their weight, there is a loss of 3.3 kgs. And we could also see that the weight loss is continued for another 3 weeks. You take one dose, and on day 22 it's another 3 kgs weight loss. It's similar to what we had seen on day 8. So this is the significant aspects when we compare in terms of weight loss what was known. And if you see that liraglutide study on 2 months every day or 3 months, and with what we had seen, we see about 3.3 kgs and it requires 2 months of treatment of 3.37 kgs. So we are trying to address with one single dose the effect of what was achieved with liraglutide. Of course, they are different population, different set of things. Just to give an example. And what else we see that there are about 11% of the subjects of more than 5%. That's what FDA would look into. Your product should induce at least minimum 5% of weight loss, and it should be sustainable at least for a week. And so we also see significant triglycerides with one single dose. How much we see that? We see about as much as 40% depending on the baseline. This intermediate 2000 microgram, they had about 163 milligrams per deciliter. There is a significant triglyceride, and it continues to show a reduction on day 15 too. Got stuck. Yeah, about as much as 20%. So you take one dose, you have 40% of triglycerides, and most of the subjects are within the limits now. So that's the kind of an effect, not only on the weight loss and also as well as in the triglyceride lipid levels. Can we have a next slide? And there are subjects of more than 150 because your the upper level threshold is 150. If you see all of them, they straight away go to 74 milligrams in 1 week time. Next slide. So if you look into the fibrates which are used for the reduction of triglycerides, it requires at least 24 weeks to have such a kind of effect, 42%. And what we are seeing, 40% in one single dose. So that's the magnitude of an effect which we see with our product in terms of treating a patient with hypertriglyceridemia. Next. So what we have a product with T-half. I said that when you have a product, it should be having a reasonable T-half. We have about 100 plus hours. So that means it's a treatment for once a week, and it's also dose proportional. So you increase the dose 1500 to 2000, 2500. You have dose proportional changes in terms of the PK properties. Next. So we also did, studies in the multiple ascending doses and two cohorts, and 680 micrograms, and the other cohort doses every week. If you look at the next slide, you see that there is a glucose reduction. We challenged that we want to understand what would be its potential to, extent of the glucose reduction upon challenging it oral glucose. And we see that 58 milligram over deciliter of glucose control in both the doses, 44 the other dose. So that means you are able to control the amount of glucose in circulation compared to the patients who did not receive the treatment. So there is an effect in terms of the glucose disposal with our product. It also indirectly suggests that you would have produced sufficient amount of insulin to take care of the glucose excursion. So that's an indirect evidence that it could be treatable for type 2 diabetic patients. When there is an excess of glucose in circulation, you would be able to address that glucose control. Next. But what we had seen, the insulin levels reduced compared to the day -1. It's not that it's increased. So what it did is that the insulin levels being sufficient to take care of the glucose excursion, so it sensitized the tissues to uptake of the glucose. So you don't need no more of excess of glucose, that your pancreatic beta cell health would improve over a period of time because your molecule is not asking to secrete insulin. So that's the another benefit. You don't need to secrete large amount of insulin and give a stress to pancreatic beta cell when you are treating. So you see that reduction of insulin levels. We also see that HbA1c level, they are normal subjects. We never seen hypoglycemic episodes in these patients, but we still see the subjects with 5 going to 4.9. That's the another aspect you don't see with any hypoglycemic events, but still some levels of HbA1c control, which is significant. Next. Once again, the absolute change in body weight about 4.6% and 2.9% at different doses. Next. So what we did, this was developed in India, everything. We did everything in 30.5 months, and we filed IND. The left, the first in human, we are able to demonstrate in 30 months. So it's not that we require several of the years, but if you focus, and we would be able to do things, and we have all of those skills and understanding, and we should be able to deliver things, and we can be. Next. So these are the competing things. We need to see that where we would stand with compared to semaglutide and liraglutide. Next. And if you see that the company which produced semaglutide, Novo, that company's the market capital is much larger than the Denmark's GDP, the country's GDP, one company market capital. So that's the power of innovation. Unless you have the power of innovation, we would not be able to command the price what you want. Next. And this year, Jan, that 1 month sale is about 5 billion. So that's the significance. Next. And what it can treat, it's not only diabetes, there are about 10-20 disorders. So that's the landscape one can look into. Next. And what most important is that the amount of spent in India is much less compared to any other country. And we being the most population country-wise in terms of population, but in terms of resources, in terms of educational this thing, we spend very less. So that needs to be most important. Thank you.